webinars are made possible thanks to generous donor support. If you'd like to contribute, please visit our website, autism.org.
So today I'm going to talk about maternal autoantibody related autism. And this is an area of great interest for my lab, and it's something we've been developing for quite some time. And I just have to provide a disclosure statement. I'm the founder and the chief scientific officer of a company that we started at UC Davis based on this technology called Modifiable. So I just wanted to get that out there. I don't have to tell you what autism is for this audience, but what I would like to talk about is sort of a new way of looking at autism. And I think this is something that during my tenure as a researcher came up, strongly in the last 15 to 20 years. We really started thinking about how the immune system may play a role in autism, and in some cases, but also how the maternal immune environment is really important for healthy neurodevelopment and what happens when we have an issue. So one thing that we look at is the maternal gestational inflammatory environment. And I'm sure everyone during the era of COVID is familiar with inflammation, the term inflammation, but also what that means during brain development. And that's going to be the topic. I will talk about that in a couple of weeks at my next webinar. But today I'm going to focus more on autoantibodies. And autoantibodies are antibodies that your body produces that we often produce. We produce them to protect us from anything that we're exposed to. However, sometimes the body mistakenly produces them to self-proteins, and that's what we term autoantibodies. So they recognize proteins in your body instead of just foreign proteins. And they're more prevalent in women, unfortunately. And there's a precedent for maternal autoantibody effects on fetal development. And I think the best known case of this is systemic lupus. And in SLE, women who make autoantibodies to nuclear proteins end up creating a scenario where the child develops a congenital heart block and can be fatal. So this is something that, I'm an immunologist. I've been doing research in autoimmunity my entire career, and this is a very well-known case. So I think that is what led us to the overarching goals of our program that we have, and that's to look at the roles of the environmental factors. Genetics always play a role because the genetic underpinnings of everything directs both our immune response, the way we respond to things, the way we develop. And the immune system is really integrally, it's very integral to healthy neurodevelopment, but the immune system and the brain develop at the same time during gestation. So that's also very important. And we do this through population studies. So we look at large population, human population, clinical studies. We use animal models, as you'll see today. We use these to help us model what we think is going on and to better understand it. And we address how environmental triggers and immune dysregulation can affect brain development. We also look at to determine how biologic markers, such as those related to immune system dysfunction, may play a role or may be important in helping us understand the role of immune dysregulation in autism. And today I'm really going to focus on the gestational immune environment and how changes in immune signaling molecules affect neurodevelopment, which I will address more in the next talk, and the role of maternal autoantibodies to fetal brain proteins and their potential role in ASD. So what is MAR-Autism? Well, this is something that came out of my lab, and this is work that came from quite a long time ago, actually. Maternal autoantibody related autism is sort of we call it a new subtype of autism because it's what we call a subphenotype or a set group of individuals that produce these autoantibodies. And so when you're pregnant, your antibodies cross the placenta to protect the fetus from everything that the mother is exposed to. And this is very healthy, right? Because the immune system of the fetus is still developing. It is not mature yet and can't protect itself. So as these antibodies cross, unfortunately, if you have autoantibodies or antibodies that are going to recognize self-proteins, they also go across the placenta and combined to their targets in the developing brain. And the illustrations I'm showing here is from some of our animal models, and where the antibodies get inside these developing neurons. These are very early neurons. And this is a control, and this is one that was exposed to the MAR autoantibodies. And you can see a big difference in the dendritic spines on these axons. So it does change how the neurons develop. This happens, we see MAR autism, it ranges from about 18% to 26%, and that range is really based on the population that we're looking at. Sometimes we've seen geographic differences in the percentage. We've seen a difference in women who already have a child with autism when we're testing that, and then we're continuing to test that right now to see what those numbers really look like. And so that's the higher end of that scale, is if you've already had a child, we know we're more susceptible to having a second child with autism, and that risk is about 20%, but this increases that risk even further. And MAR-ASD is really a distinct behavioral subtype. So there is, even within this subphenotype, there are differences depending on the patterns, and I will explain that a bit more in a minute. But we see more severe behaviors affected, more pronounced stereotypic behaviors, and significantly larger brain volume, and I'm going to talk about that quite a bit. Because we see that in humans and in our animal models. So the initial finding for this was a very long time ago, and the first paper was published in 2007 on this, and it all started when we took fetal brain from that we created from monkeys, and we ran it in a gel, and we took antibodies or serum from humans, tested it against those proteins, and we kept finding this band pattern. And that's really, this is the IgG, heavy chain and light chain, that was present in the fetal brain. But every time we saw this, it happened to be a mom of a child with autism, and we didn't see it in the typically developing or the developmental delay controls. And that actually was the beginning of everything. And so it's been a very long, sometimes rocky road, but we've made a lot of strides in this. And I think, as I said, we see differences in behavior, and even our most recent paper that just came out this year is also showing that, and I'll show you the data from that. We noticed that there was a genetic association with the production of these autoantibodies in a gene that regulates, or that controls inflammation regulation. We saw other factors that autoantibodies are associated with metabolic conditions in the mother, so that is an increased risk of having them. Large brain volume, I'm going to talk about that a little bit more. And we see it in all of our animal models, every single one of them. So this is something that's been consistent all throughout our studies. We've determined that we've found 8, and then Lior Brimberg and Betty Diamond found one that was independent of ours, and that there are different targets, and I'll talk about that more in a minute. We have done a lot of animal models, and the reason we do these is so we can understand what these antibodies are doing and potentially why. And so that's, this is an important part of what we do, and we've now are creating our rat model. And then we're looking at the brain tissues. And so as you saw the pictures on the early slide of how we're changing the way the brain develops with these antibodies, and I'll talk a little bit more about that. So there's a lot of us, there's a lot of data on this now, up to our most recent paper by my graduate student, and the Brimberg work is not in my lab, so this is independent of us, and it's really interesting. It's a little less specific. When I talk about our studies, I'm talking about with 100% accuracy that we're predicting if a child will have autism based on this panel of autoantibodies. We also, a number of animal models showing that these antibodies actually have an effect. And so that's, I will talk a little bit more about that. But one of the things, when we find these panels, and we've identified what all the proteins are now, they're great biomarkers. So if with 100% accuracy, we're predicting that these antibodies are associated with having a child with autism, they may not be pathologically significant, or they may not be causing disease. And so that is the other reason we use the animal models. Are they functionally causing an issue during development? And these are the 8 proteins that we determined You will never see them at this point because the antibodies don't begin to cross the placenta in humans till around day 100, day 110 of gestation. So even if these proteins are important for early events, the antibodies will never see them. So they don't begin until later on crossing. And so they are definitely, the STIP1 protein is important for neuridogenesis. Cypin or guanine deaminase is for dendritic branching. So each along the pathway, NSC is for glycolysis neuron specific enolase. Lactate dehydrogenase is in every single cell. It's in the mitochondria, but it's very high in the developing brain. And that's because the brain does not have a lot of oxygen in the fetal compartment compared to when the child is born. So LDH is important for metabolism. YBOX1 is a transcription regulator, and then CRIMP1 and CRIMP2 are kind of the most interesting of the 8, and they are really important for this later development of the axon outgrowth, growth cone collapse, and basal dendrite patterning. So they really are, these are, I think, my favorite too. They're the most interesting to me. And so what we, sorry, I thought I hit the thing twice. So the summary, so this is from our most recent paper, and we had done a number of studies, but I think we're really proud of this because this study, we developed this assay to be incredibly clean, incredibly specific, and we were very stringent in our cutoff so that we made sure that if we called something a positive, it really was positive. We were very careful and did a training set. So when we say training set, it means we determined what the patterns were that were specific, and then we do a validation set. We take a whole nother set of samples and we take those patterns and see if when we apply those patterns, we're correct. Fortunately, we were correct 100% of the time with these particular patterns. And we call these backbone patterns. And the reason we say that is because you have to have at least these two. So CRIMP1 GDA, CRIMP1 CRIMP2, and NSC STIP1 are the most common patterns that we have and the most abundant. They're all 100% specific. So if you have any of these combinations, but very often people have more than just two of these antibodies to more than just two of these proteins. But if these two are in the pattern, then it will be, it predicts that the child will have autism. So the odds ratio mean here means we're 31 times more likely. And the reason that it's not 100, because I was saying it's 100% accurate, is just based on the numbers that we've tested. So as we accrue more numbers, that number will change. But we were really excited by this, and it verified what we had found prior to this. And so I think, this allows us to create what we hope will be a way of creating a biomarker. And my cat has chosen this moment that she needs attention. Sorry, the joys of working at home. But one thing that we did notice is that so we did another study, a small study, and this was what I was talking about when I say we had geographic differences a little bit. But what we noticed is that if they are MAR positive, so these are the MAR negative, you get a sort of a normal distribution. If you look in the MAR positive, they're all on the ADOS severity score 6 or above. So a lot of them at 6. So we noticed it was shifting sort of the range to a more severe phenotype in these kids. And same with the SEQ, the social questionnaire. So say again, sort of a normal distribution in scores where we shifted things significantly to a more severe outcome. And the other thing that we noticed was that CRIMP1, antibodies to CRIMP1 seems to be driving some of the severity. And remember, CRIMP1 is in the CRIMP1 GDA pattern, and CRIMP1 CRIMP2 pattern are the two most significant patterns that it is part of. And what this means, this point estimate, it means that if this pattern is part of the autoantibody pattern, the ADOS severity score will increase by an average of 2.3 points. So if it was a 6, it might be an 8.3. So that's what that's telling us, that it is part of what is driving that increased severity with these autoantibodies. So as I said, and I see we have questions coming up, that's great. I wish we could stop and answer them now, but I want to make sure we get through things, so hang on to those. As I said, there are modifiable risk factors associated with these. It looks like there's an increased prevalence of these autoantibodies in mothers that are diagnosed with diabetes, hypertensive disorders, or that are overweight or obese compared to healthy moms, and this is during pregnancy. Mothers of ASD severe children and diagnosed with type 2 or gestational diabetes were nearly 3 times more likely to have the autoantibodies compared to healthy mothers. So this is a modifiable risk factor, right? This is a factor that hopefully we could potentially control at least, you know, one risk factor associated with this. And the cat is just not giving up here, sorry. So as I said, are these autoantibodies pathogenic? And what I mean by that, do they cause damage? Do they cause harm? Do they change the way the brain develops? And that is, you know, certainly what we really want to understand is, and we established that they induce the maternal antibodies induce autism in our passive transfer models. And so that means we take human antibody and put it into the animal, and I'll show you some data from that. We show that they localize to the brain in some of these studies. So that's another question, right? If they get across the placenta, do they actually get into the fetal brain? And then we created clinically relevant models where the dams produce the autoantibodies. So now we make the females produce them. We don't passively transfer the human antibody in anymore. And this is what we call an endogenous model. And it mimics, we created it to mimic the reactivity that we see in our clinical samples. So the passive transfer models are where we take human IgG that are reactive to the target proteins and we passively put it into the animal during pregnancy. This is so I'm going to show you some data from one of our non-human primate studies that I did with Melissa Bauman at UC Davis. So she's a very good behavioralist. And the rationale behind this was that human behavioral phenotypes correlated with the autoantibody pattern. And in the early studies, lower expressive language and increased stereotypic behavior were definitely associated, with lactate dehydrogenase CRIMP1 and STIP1. So that was really our early big pattern that we found. And we transferred 6 times, beginning late in the first trimester and into the second trimester, into pregnant rhesus macaques. And this is a very slow study. First of all, this is a very expensive study to do, but it's also very slow in that you have to, the gestation time is long. It's not like a mouse or a rat. And then we have to wait till they grow up enough to be able to do the behavior assessment. So it's very similar to what we would do. The case in humans. And interesting data came out of this, one of which was that the mothers of the IgG treated monkeys, so these are the ones that got antibodies from women who had IgG from women who had these autoantibodies, were overprotective, which is sort of they didn't let their offspring go off and sort of explore as much and interact, and which is also going to change their behavior, right? That's going to change their behavior development, their social behavior. As they got older, the IgG-treated offspring showed evidence of abnormal social behavior with both familiar and unfamiliar pairs, and I'll show you some data from that. And it's kind of interesting when you do non-human primate because they're so much more complex than when I show you the videos from mice. Mice are pretty simple, but they have a number of facial expressions and ways that they approach. And so the primatologists that are really good at this behavior notice that they might approach another animal, but they were exhibiting behaviors that the other animals were not comfortable with. And so they didn't get what we call species typical reciprocal social response. And so in this graphic of that, you can see, so this is the number of behaviors that they initiated, meaning how often they initiated a behavior. And this is affiliative behaviors that were received, meaning how the reciprocal of that approach behaved. And you can see in the control animals, they and over time, they would, as they initiated more behaviors, they got more reciprocal behaviors back, right? So there was a back and forth between them. And in the MAR-ASD IgG treated animals, it actually went the other direction, because there was something that was not seen as like a normal engagement process. So we also had the ability in this study to image these starting very, very young at 1 week of age, and we could image these offspring over time. And on the top, these are the IgG from the MAR-ASD, and on the bottom are the control animals. And that we saw an enlarged, elevated growth trajectory, and they had larger brains throughout the time that we were imaging them. So this is something that was really interesting, because back in 2013, before we published this study, we did this study looking at imaging, just this is total cerebral volume, that this is the ASD population on the left here that did not, the moms were not positive for the MAR antibodies, and this in the middle are the ones that were positive, and then this is the typically developing controls that they imaged. And you can see there's quite a range in both of these two groups. You have some at the top up here and some down at the very bottom in total brain volume. But here you can see it's very tight, and we have the largest brain, these are all boys, the largest brain boys were actually from the MAR-ASD moms. So this, this is now we're replicating what we saw in our animal models, and I think our animal models are replicating what we saw in the humans. And I think this is always a good thing, right? So it means we've got some kind of a physiologic effect of these antibodies on how the brain is growing. And then we really wanted to look to see if these antibodies were getting into the fetal compartment. So we knew that we saw that enlarged MRI. We saw the same thing in mice, and I'm going to talk about that in a second. There was effect on titer, antibody titer, on behavioral severity in our animal models and one of our passive transfer models. So, and then we used PET imaging, where we labeled the IgG from a human IgG, and on embryonic day 14, we injected into the dam and tracked where that antibody went. And even so, on this is prenatal day 19, which is 5 days later, we can see there is there's this light spot in the brain, but you can see it's actually throughout the fetal compartment, very high concentration at the placenta. And so this is something that we're really interested in, is how this might be affecting development through effects of these antibodies on placenta. But even 12 hours after birth, so this is a number of days, there's still there's no difference between the controls and the MAR animals, because IgG is in both compartments, but are in both animals equally. But there's still a lot in the brain, even 12 hours after birth. So it gets in there and it's staying in there. So it's not a transient event. It continues to have an effect. And then this is some work that I did with Veronica Martinez-Odeño at UC Davis. She's a neuroanatomist, she's an excellent neuroanatomist, and she actually injected in these tiny little heads on day 14 of embryogenesis. And we had them, the antibodies were labeled so we could track, and it was injected in the space that's white down here below the brown, into the ventricle. So this is a very artificial, this is not a model that you would, want to take put a lot of stock in for how this happens over time and outcome. But what it did allow us to do was look at what happens if we label these antibodies, where they're going. This is a control. This got control human IgG. As a matter of fact, we did it a couple times because we thought we hadn't gotten it labeled correctly, but it was labeled. And the difference between these two is these antibodies, when an antibody finds its target, it binds with very high affinity to that target. And even through all the washing and all the processing, it doesn't come off. And so it's also getting inside these cells. And these are early developing neurons, and it's changing the way these cells behave. It is not killing them. It's not wiping them out. We don't have less cells. What we actually have is higher accelerated migration of these radioglial cells. We had accelerated mitosis, meaning they were dividing more frequently. This is a control over here. You can see the radioglial cells. They're where they should be, but at the same time point, these look very different, and this is graphically depicted over here in the number of cells in each bin that they looked at. So how they're taken up into the developing neurons is a big question for us. Some people say that, you'd have to, for an antibody to be effective, it has to be against an extracellular. And that is certainly was the dogma that I was trained under, but I think we're sort of changing how we think. And I think there's a permissive point that these antibodies can get inside these cells. So my graduate student, Matt, is working on this to better understand that. But what we did see when we, in the animals that we didn't use for this staining, in the adult animals that were treated the same way with the intraventricular, we saw a big difference in how the mature neurons looked. And so these are adults, and meaning they've already gone through development, but you can see the ones that were treated with the typically developing IgG and maternal IgG, lots of dendritic spines, they look totally normal. This is the ASD, and these are the ASD, and you can see they look vastly different. Even I, who am not a neuroanatomist or neuroscientist, can tell that there's a big difference in how. So these little spines are what create your connections between neurons, right? This is your communicating piece of the neuron, and so if those are absent, there's no communication between this neuron and another neuron, or these are very rudimentary down here, these tiny little ones, they aren't fully developed. So I think for us, this was really interesting, but we needed a better model. And I think this was a very pivotal point for my lab in terms of creating an animal, but the dams make the antibodies. We used the clinical epitopes. And what I mean by epitope, an epitope is that little piece of a total protein, that peptide that the antibodies recognize. And the important part of that is we needed the antibodies that our mice made to look like the clinical antibodies. Otherwise, it would be useless to us. We started again with the LDH, A and B, STIP1, and CRIMP1, because that was our major pattern at the time. And now, when we create them in the dam, so we immunize the animals before we ever even breed them. We make sure that they're making a really good titer to these little pieces of these little red dots, or that's how we epitope mapped. And then these antibodies are now present all throughout gestation and lactation. So they're there the entire time. And that was a really important aspect too. And then we did early postnatal behaviors, juvenile series of juvenile and then adult behaviors in these animals. So one of the things that we saw was altered neurodevelopmental trajectory with the total cerebral volume. And this, even if we just take calipers and we're measuring their heads as they develop, we could see there was an increase in growth in males and females. So this point is important later. We do not see behavior differences between the males and females. I know autism has a heavy male bias, right? 4-to-1 ratio. However, with this type of autism, it doesn't, it may look a little different, but they both have similar outcomes. And what we saw was the pink areas are highlighting all the places that we saw elevated increase in total cerebral volume here in these various spaces. This is an endpoint experiment. So these were adult animals. So this is what they look like at the end. And this is in collaboration with Jacob Ellerman and Jason Lurch, who are the brain imaging up in Toronto. But what we did see, which was interesting, it was a lot of this total cerebral volume was driven by the females, which is completely opposite of what we thought we were going to see. They both see an increase, but the we see a bigger change. And you can see there's actually the female brains are a little bit bigger at this time point that we were looking at the adult time point. But the significant difference was really between the MAR females, the female animals. Now, as I said, we saw behavior changes the same, whether they were male or female. So it is really interesting for us that there's a big difference in how the male and female brains respond to this exposure. And so we're continuing to look at that. But there is a relationship between the behaviors of the MAR male. So the MAR male mouse has the number of increased grooming. So self-grooming is a reciprocal or repetitive behavior in a mouse. Oh, God, my old dog's coming down the stairs now. And the control males, you can see there's not a difference there. And so there is what we saw with, even though the females had bigger brains, there was a relationship between the brain size and the severity of the behaviors. And so this paper is in revision. We just sent it back to the journal to get, we've revised and sent it back to molecular psychiatry. So hopefully this will come out soon. There's a lot more complicated stuff in there, but I just wanted to sort of share that, we may see a finding, but the sex differences, we really need to look at the male and female animals to truly understand what's going on. We can't just look at males and females as if they're the same. And I think this is a trend we're seeing just in science in general. So this is a technique that we use called clarity imaging. And this is where there's actually a brain in there. You can barely see it in this test tube. And the brain is cleared from sort of the fat. So you can image through the whole brain. So you can image the entire brain, and it enables you to look at a 3D rendition of what the brain looks like. So we did this on the mice that that we were just talking about. We took the postnatal day 2 mouse brain that was exposed to these antibodies, and we stained just for anti-IgG. So all we're looking for is where the antibodies that the dams made, right? So now that the dams are making these, not us, we're not doing anything except staining to see where these antibodies are. So on the left is an animal that the dam made the anti-LDH crimp1, STIP1. On the right is a control animal that had saline and adjuvant that didn't have any of the proteins. And what you see there are the blood vessels. Normally we perfuse and the antibody would have been cleared out of there, but apparently it wasn't perfused as perfectly as it could have been. But perfusing, washes. And so after washing, these antibodies, remember when I said that the antibodies will find their protein and stick to that protein, right? And that allowed us to get this image. So it's bound to its protein. And as you can see, it's inside the cells, and there's even some cells here that are dividing, and it's still inside. This IgG is still inside those cells. So to me, that was a really exciting thing, that the IgG is clearly staining intracellularly. We're seeing it even after all the washing and everything we're doing. We're still seeing this IgG bound to its target proteins. So the behaviors, as I said, there was no difference between males and females, so they're all lumped together here. So we always check that statistically. So what you're seeing is a graphic depiction of the different behaviors we look at for social behavior. And you can see we have the control animal, which has the yellow dot on it when it shows back up, and the stimulus animal. So this is where we've got these are juveniles, 2 young animals put into a cage together for the first time that they've ever met each other. And they explore around the cage because it's a novel environment, but they also kind of crawl all over each other, hang out together. They're interacting. And then you're going to see a big difference here in our MAR-ASD mouse, where this is the MAR-ASD. I didn't even have to put it. You can see that animal is what they're doing. It's just sitting there self-grooming. And the stimulus mouse keeps trying to engage the animal, and it actually actively goes across the cage to avoid that interaction. So the So the stimulus mouse tries again, and it tries to go across the cage. It tries to go across the cage, engage that mouse, and it again actively avoids. Get her. Hang on a second. My husband's going to remove the dog that is sitting here. So we were really excited by this, and these are the data that just really sparked the interest that these are pathologically doing something, that these antibodies are clearly causing the brain to develop differently. This is a stereotypic behavior cage, so they're put in this empty cage. I'm not labeling this one, so you guys can guess which one it is. That is the MAR-ASD mouse versus the control mouse. But the animals were allowed to actually explore the cage before we started videotaping. And you'll probably guess it's the one on the left, because it's just sitting there self-grooming while the other one's exploring around. And it's not that they have anxiety. They don't have a high rate. We tested that. They're not afraid to go around. It's that they just get into that place where they just get into their repetitive behavior. And then the last thing that this was one of my favorite outcomes, because this is where we do ultrasonic vocalization, and we often do it in pups, where we take the pup away from the mom and see how much they vocalize, and quite often it's reduced in these models. But this is where an adult male is put into a cage with a female that's in estrus. And this is a very exciting moment for this male, because this female, is ready to breed. And you can tell from the control voice print, all these little black blips are where the male has vocalized in excitement. And you can see it's hot and heavy at the beginning here and kind of tapers off. This is the MAR-ASD animal the voice print was taken at exactly the same time when they were first put in the cage. And you can see the big difference. And it just never really, there's just not ever much there. The stuff at the bottom is actually the cage, is movement in the cage. So, and you can see graphically here that the MAR-ASD it might have a little bit at the very beginning, but it just drops off compared to the controls. And then again, graphically here. So that is not communication like we think of communication. It is a means of communicating, or at least interest in communicating with the animal that's in the cage with them. So we've moved on from mouse, and a couple of reasons. The rats, we've moved on to rats, partly because rats have a much more intricate social behavior paradigm. They're much more social. You'll see in a minute when I show the video, they play more, they're smarter, they can learn better, and they have some prefrontal cortex, which helps make them a little bit better model. They're bigger, so it's a little more expensive, but we've really shifted away from doing the mice now and over to the rats. Now, fortunately, we got the same outcome with the rat, which is always good. We got deficits in social interaction, including reduced social interactions, reduced play. They play when they are together, when they're juveniles. I mean, they roll around the cage. They're very cute. Increased repetitive behaviors, which rats typically don't have a lot of repetitive behavior, and reduced ultrasonic vocalization in the pups. So, but this assay that I'm just have here as graphically depicted is an assay we developed at Davis called a social motivation. And this is very preliminary data, so we're expanding on this now. But the adult MAR animal is placed in both the adult and the and control offspring of the MAR animals are placed in this area where the so the stimulus animal is put into this confinement tube, and it's got a little lever here that the test animal can push to release the animal. So when they're going to put in there, they're going to make noise. They're not happy being in there, being confined, and so their peer can go hit the lever and release them from this. And you can see graphically here, and we do this several days, and they should learn the task, right? They should get quicker and quicker and quicker at it. So this is the time it takes them. now the poor people doing this task, you can see they're sitting there for 20 minutes and they were doing this for 12 days straight for all these animals. But this assay truly, you can see the division between, here's the controls, they get faster at it over the time. Here's our MAR-ASD animals. They're just like not super interested in learning this or doing it. And there's nothing cognitively wrong with them based on our other battery, but they just really, it is like releasing that, it's almost like an empathy task, releasing the animal. It doesn't occur to them that that's something they should be doing. So we're continuing to do this. My new graduate student is learning this task. And I think I'm really excited to see where that goes. This is the rat behavior I was talking about. So the same as the point of the HA&B. And these are juveniles, and they look huge, but they actually are young. And you'll see when we put them in the cage that we've got a stimulus rat and we've got a control rat here. And you can see they're so fun to watch. I mean, they just crawl all over each other. We've not sped this up. This is how fast they're moving around the cage. And we got a little mounting behavior there. I mean, they're rolling around and clearly hanging out together. Now you're going to be able to see the MAR-ASD rat, and we've got, again, a stimulus rat. And I'm not going to tell you which one it is at first because it becomes sort of obvious in a minute which one it is. And the interesting thing about this is that it's the rat in brown there, is that the stimulus rat kind of gives up. And this is what we saw in our monkey model, where the MAR-ASD rat is actually just sitting there licking the bottom of the cage at the end of this. And this guy is just like, let me out of here. But you can see the graphically very robust play behavior as juveniles, which goes down as they age, right? They don't play the same anymore. I mean, they have other social interactions that are different, but it's always lower with the MAR-ASD animal. So very happy to replicate our mouse data with our rat data. And one of the things that I wanted to bring up is, what do they all have in common? And as I alluded to, the enlarged brain is now common. So we had the human data I showed you. The rats replicated the mouse data perfectly. We actually did 2 time points in the rats over time. Here's the mouse data I showed you, and here's the monkey data. So now the one common thread through all of this is we are changing the way the brain develops in these models in such a way that it actually physiologically changes what the brain looks like for the total cerebral volume. So that I'm just going to end there. I mean, so I just wanted to, for the conclusions, that gestational immune dysregulation, we think, contributes to the risk of altered neurodevelopment. We see this in schizophrenia models. We see it in our autism models. And it may be an underlying mechanism. Something, we might link to infection during pregnancy, for example. Not that you get an infection, but how you deal with the infection. And I'll talk about that in my next webinar. And then the in vitro studies demonstrate the effects of inflammatory cytokines in neuron health and development. I'm going to talk about that more next time, but we do know that we've got sort of this immune basis for changing outcome. Antibodies localized to the brain of the developing animal, and the monkeys and the rodent models, we're seeing this over and over again. We see enlarged brains in the humans, mice, monkeys, and rats now. I think we're going to stop with those 4, but this is something that then we can, and my student is putting together the rat paper. We have a lot more data on the rat. We've got data on the what we call MRS spectroscopy, which is a very fancy way of saying we can see what the metabolites in the brain look like, and there definitely are differences. So stay tuned for that. And we see a difference in both our mouse and rat models in 3 different domains, the social, communication, and repetitive behavior deficits that we associate with autism. So we're very excited about that. And so they alter these antibodies, we now know, change the way the brain develops. We see this through the cerebral volume. We see it through looking at the neurons themselves, how the neurons are migrating in the brain, and seeing these structural deficits. So this is my gang of people that thank God work with me. Melissa, I talked about Paul Ashwood, who's been with me since he was a postdoc, and he's now an independent investigator at the Institute. But Paul and I work together a lot. on these. Dr. Lisa Crowan does the epidemiology human studies with me. And then Ale did the paper on the, that we created this assay to test for these antibodies. Matt and Jana are doing the rat model, and then Danny is working on the inflammatory side of things and creating our biomarkers for inflammation. With that, I will stop sharing, and I'm going to go into the chat and see what we have and the Q&A. So I'm going to start with the Q&A and see. I'm kind of trying to get through these, and I'll have Denise can check as well. But I think so. I think one of the questions is, if you have other autoimmune disease disorders, does that impact the outcome, right? And Lisa Crohn and I actually did a study on that, and really it was the autoimmune disorders that are really T-cell mediated, like psoriasis, psoriatic arthritis, type 1 diabetes, sort of some of those disorders versus just generic, and all asthma and allergy. We didn't see as much with those that had other autoimmune diseases. Now, that said, if you have an autoimmune disease, it means you have an issue with immune regulation, right? You're not regulating inflammation as well. And so it is possible. It is possible that by virtue of the fact that you have an autoimmune disorder, you are at higher risk just because of the inflammation that goes along with that. So I think, you know, I wish I could answer. The question is, you know, could it be happening? She's someone that texted a question that it is, you know, if you're pregnant now, and I think if your autoimmune disorder is, you know, regulated and under control, it should not have an impact. That's not to say that there might not be something else related that we don't know. And I wish I had more information for you. You know, these are things that we're just still really learning. So people are asking, like, can we get tested? So we're not testing anybody during pregnancy because of for the obvious reason. We don't have to, because it's not, this test is not related to the fetus itself. It's all maternal. So it's the maternal environment. So we could test prior to gestation or early postnatal. And so we're working on getting this test commercialized, which is when I disclosed the company that we're building, just so that I can get this back out to the public. So let's see, are the research studies now to track moms with an ASD child during pregnancy and monitor baby SIBs? Yes, the M.I.N.D Institute has the baby SIB studies are going on all over the U.S. So those are important studies. Is right that yes, Marble study at Davis, some of the ECHO studies, the early study. There's a number of studies that are doing that, but the Marble study is the one we're part of. And so would an immune modulator, like before and during pregnancy, lower the risk? That's a tricky question, because we don't know what other effects those drugs have. So I think, you know, before we start just blanket doing that, and I realize it's really frustrating to have to wait for the answers to these, but I think we want to be very careful on what we say to do or not do if you suspect inflammation. I do want to point out that, and I will talk about this more in the next webinar, but I think what I really want to point out is that a lot of people get sick during pregnancy, and a lot of people have healthy kids afterwards. So what we're trying to understand is sort of the other things that are associated with that push that inflammation into a more chronic state, or to a level that's, you know, above a threshold that then tips the developing fetus into maybe a neurodevelopmental disorder. If a woman has these autoantibodies in one child with ASD, what is the risk of subsequent children, especially males, will also have ASD? is it basically unavoidable? I think, is it unavoidable? If you have the autoantibodies, you know, I'm sorry to say that yes, that are, at least from every study that we've done so far, that the likelihood of having a second child is very, very high. The risk is already 20%, so higher if you have a child with autism, that you will have a second child with autism, unfortunately. IVF, using a donor embryo, you wouldn't even have to have it be unrelated, but using a surrogate, right, to gestate would remove, in the case of the autoantibodies, would remove, you have to test surrogate, but would remove the environment of those autoantibodies. We're working on blocking technologies so that we can, you know, block this during pregnancy, because you only have to block for a short period of time. How can a woman prevent having these antibodies beyond the obesity component? You know, that is a great question. It's very difficult to know when we don't know what causes them to happen in the first place. And this is my entire career in autoimmunity. You know, we don't always know. We don't know what the incident was that drove the loss of self-tolerance to the point where you're making autoantibodies and then have an autoimmune disorder. But how you prevent it, obesity, I think when I say obesity, and I think healthy, obesity is linked to inflammation. So you want to have a low inflammation environment. I mean, that's just healthier for all of us anyway, to have, you know, not have this sort of, you know, inflammatory environment in your body constantly anyway. I think we're preventing the autoantibody production, and is a trickier answer than can we do something about it when they're there, and that's kind of that direction that we're going. Well, thank you for the comment. Thank you for my easy and understandable explanation. I try. I'm constantly trying to use different words than immunology words that we just throw around. And the question about, is there an association between the autoantibodies and pro-inflammatory cytokines? Excellent question. My graduate student, we've got another study where we looked at these antibodies during pregnancy and saw the same patterns. So that's a paper she's preparing. And then the next link of that is we've also got the cytokine profiles from those same women taken during pregnancy that we see if there's a link between them. And that will be definitely something that we'll look at. And then, so Lurie Center has found immune activation from postnatal exposures. Are we looking at this exposure timing as well? Yes. I mean, we obviously, I'm talking today about, you know, what's going on prenatally. We absolutely look in the kids at what's going on. That's where the intersection of Paul's in my lab is definitely at the postnatal. We look at GI, we look at it, this is just a part of what we do in the lab. And so we definitely are looking at, especially in the context of environmental exposure. So I'm putting together a paper now that will, I think, be interesting in the context of that. Let's see. Oh my goodness. I'm trying to get through them. How you can get tested? Stay tuned. We're like, I hope, you know, sometimes it's a little bit of a financial question of getting this test. We're moving this test from my research lab to a more commercial setting. And that's an important part of that, because the assay validation at the commercial setting, the clinical studies that are needed, there's a whole nother layer before we can take this out to the public. So we want to be really careful that what we have is real. Speaking of our ASD, are they at increased risk for having adverse reactions to vaccines based on inflammation antibodies? I think the adverse event risk is something that we're looking at actually more in the context of our maternal immune activation model in the non-human primates and in the mice, which we think is actually a little bit better model for schizophrenia than autism because of the onset and the pattern of onset and the behaviors. But does prenatal maternal immune activation affect how the child's immune system develops? It does. Does it make it so that the child responds more poorly or, you know, has too high a response? to an exposure, that is the question that we're addressing. And absolutely, you can email me if you have questions. This is my favorite part of giving a talk. I was trying to get through my talk so we could have more time. Is this for any child, whether mild or severe? Yes, because one of the patterns actually is milder. One of the patterns. I was talking about what was more severe. I would have had more data on that if I'd had more moms with a given pattern. So that's why we keep going. Various avoidant and supplemental interventions have shown benefit in mitigating risk. I'm not sure what Dave meant by that, but yes, there are, unless I'm not seeing the whole question here. And I think one of the we're mindful of is absolutely what we're exposed to. And I'm going to go to the chat room now. So that was the question and answer part. So let me get back to the top. Is the diabetes a causal reason for the increase or a symptom of another problem causing both? That is a really great question. I think we know that there's a risk from gestational diabetes in autism to begin with. And we know that gestational diabetes, obesity, all those are inflammatory. And is it a combined effort? So this is something that we're trying to understand, right? And we may have to create, there is an obese mouse model. And if I combine the two of them, I'm almost afraid to look at what those poor mice would look like. But I think, this is a way we can potentially understand that. The question was, this new biomarkers will be a new way to diagnose autism. We hope it's a new way to get at an earlier diagnosis. I mean, we're very good at behaviorally diagnosing autism. I think, we've got that, I mean, the people I work with are incredible at it. But I think, will we be able to do this before our standard? Yes, I hope that's the hope, that we'll be able to do that. And are we gonna, yes, this has all been recorded, so you can, somebody asked if we were gonna replay. Yes, there will be. This whole thing has been recorded, so you can watch it again, and I apologize for going so quickly. So among the subset of autism cases with MAR, the male to female ratio, prevalence ratio, well, in the samples that we were testing, the prevalence ratio was 4-to-1 male to female, so we had fewer females, but there was no difference in sort of the normal ratio. It wasn't like a ton more males had it than females. As I said, this is an equal opportunity mechanism, because it's not, the issue is about the gestational environment. What are the possible treatments for our kids? That is a really important question. We did some downstream RNA-seq, which is a fancy way of saying we looked to see what RNA message was being made in the brain that had been exposed to these antibodies during gestation. And so we have a number of targets that were changed, some up, some down compared to the controls. We can look at those because that now is reflecting the kids, what's going on in the kids themselves, that we potentially could target, we hope. And I'm sorry, I wish that things were faster. I do. I wish we could get the test out faster. I wish I could get an answer faster. That is the most frustrating part of my job. But we are working on it. And so let's see, are there any labs? Are all the labs of it? No, they are not. So there's no place to get this test until we create. As a matter of fact, in half an hour, I have a call with my team to look at our clinical use right now. I mean, that's the question we're answering, and how we're going to get that done. My mom and aunt have autoimmune disorders, but their children are not. ASD, including me, but my children are. What is different? Oh, that's an interesting question. Just having an autoimmune disorder, as I said, is not necessarily a risk. It depends on what kind of autoimmune disorder, how well it's controlled. We inherit sometimes the ability to have an autoimmune disorder ourselves from our parents, but sometimes not. Just like some of us get asthma or allergy from our parents that have it really badly, and some don't. And that's all all the genetic recombination occurs. Just sometimes you get lucky and you don't get that outcome. And I think, did I get through all the questions? Dave is sending me messages that thank you, Dave, for putting your comments in. I say I had no gestation. My son, so could it be that there was some form of neutral inflammation during pregnancies? I think, I'm sorry, I don't have the answers to all the questions because there's a lot we don't understand. And I know that's like a cop-out answer, but there really, sadly, isn't, like, in terms of cause and effect. And all I can say is if reducing our inflammatory load is I think important for all of us. Myself as somebody, what I call inflammaging, which is inflammation as we age, our inflammatory levels sort of go up. Trying to control that as best we can, especially through diet, is important. I mean, just for our healthy best self. Certainly during pregnancy, I think it's important. But I think also not stressing and not panicking is also key. I think that we know, like, being just super stressed about it actually also has a negative effect. And I know that's a very easy thing for me to say. My child's almost 22 now, but I think, that is, we publish science at the best our ability to get you the best information we have at the time we have it, but always know that it's the best we could do with the tools that we have, and it does not always apply to every single case. And so, but we're trying to get you the best we can at the time.